The New Hampshire section would like to welcome Dr. Vikram Pattanayak, doing a presentation on an overview of CRISPR and Cas9 and gene editing. Dr. Pattanayak is a professor at Harvard Medical School. He's the director of the Histocompatibility Laboratory at the Massachusetts General Hospital, and as I mentioned, instructor in pathology at Harvard Medical School. So I'll turn it over to Dr. Pattanayak about CRISPR and Cas9. Thank you.
Thank you. Great. Thank you, Jim, for the introduction, and thank you, Shri, for the invite. Really excited to give this talk, and it's very timely if you've been following the news. So I'm talking about CRISPR, Cas9, and gene editing. I'm going to try to, at least at the start of the talk, give a little bit of an overview of DNA and biology a little bit, so that there's some framework for what I'm talking about. But I understand that, some of you will have heard of DNA and gene editing, and some of you will not have. Before I really get started, I'm required by my institution to put up this disclosure slide. So I hold license IP related to the content of this talk, and I'm a co-founder of a company, SecureDX, which is a company developing technologies for gene editing, target profiling, which I'll be talking about briefly at the end of the talk. And these potential conflicts of interests are managed by Mass General Hospital and Mass General Brigham in accordance with their conflict of interest policies. So right now, this week is actually a really special time in the gene editing field. But for me, my personal introduction, and maybe a lot of you got introduced to genomics in a major way back in 2000, 2001. So I was a freshman in college when these two issues of Science and Nature the same week came out, announcing the completion of a draft of the human genome, which was a project that took many years, lots of technological developments in both the public and private sector. And it's amazing that this was actually only a quarter century ago, essentially, that this all happened. And when this happened, there was obviously a lot of hoopla. And you might remember President Bill Clinton and Francis Collins and Craig Venter in a news conference touting how transformational this human genome sequence might be. So Bill Clinton mentioned that humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. When he said that, I don't think any of us could have actually envisioned that just last week that the FDA in the United States would approve a therapy that actually changes some cells in a patient's that you put into a patient. Their cells change it and edit a gene sequence. So the human genome, a quarter of a century, kind of completed the definition of all the human gene sequences. And what Bill Clinton was talking about, revolutionizing the diagnosis and treatment, I believe what he meant was understanding how disease happens and what the hereditary or genetic basis of disease is, and what proteins might get mutated in the course of somebody's life that can cause diseases like cancer. And certainly we've learned a ton about this, but I don't think anybody really envisioned that we might be editing genomes. So you might be asking what a genome is, and I didn't put a clear definition of genome. But people and organisms are made of cells, and in each cell is a genome, which is 3 billion base pairs or letters of DNA which carry information. And the information carried in DNA, according to what's called the central dogma of molecular biology, is transited through a messenger RNA molecule before getting translated into protein. So DNA is an information carrier, and protein is kind of like the functional output of that information. So you can think of DNA as blueprints for, a functional entity, a functional molecule. We can represent DNA as a string of letters, and we can also represent proteins as a string of letters. These literally are strings. So each letter is subtly different, but there's a common kind of backbone that ties all of these letters together. So for DNA, the letters are called nucleotides, and on the nucleotides there's bases, which I'll show you in a second. But those can come in four varieties, essentially: A, C, G, and T for adenine, cytosine, guanine, and thymine. Proteins come in 20 or 21 different flavors depending on who you ask, and so we have to use more letters of the alphabet for the proteins. The way our genomes and cells are structured is that as the information carrier, the letters of DNA directly correspond to the letters of the protein. So the DNA nucleotides correspond to amino acids of the protein. So each of these letters is a representation of an amino acid. So sticking with the letters for now, this is the code um, that took lots of effort to solve half a century ago, more than half a century ago. And you can see there's a correspondence of 3 letters of DNA to 1 amino acid. So these are, instead of single letter abbreviations, these are 3-letter abbreviations for the word. So phenylalanine, leucine, isoleucine, alanine, aspartate, etc. It's easy to represent DNA as letters. Protein, we usually don't look at as letters, but for the purpose of the code, it's kind of easier to show it to you like this. But just know that behind the letters is actually our actual, chemical molecules. So DNA has this negatively charged phosphate backbone. So here are the phosphates, and each phosphate is attached to a sugar, in this case deoxyribose, and this is invariant. So let me get my pen. Um, the part uh, the part of this molecule that's to the left is just a repeating unit of these phosphates and sugars. What differs is these bases. So for instance, in this example, this is a G or a guanine. This is a T or a thymine. This is a G again, guanine. But so you just have this repeating units. But the bases are different, and that's how you encode information content. And if you think of Shannon's law of entropy, you can actually encode a lot of information in DNA. So this DNA, once it's copied into RNA, which is very similar in structure, and then translated into protein, it starts to have this kind of structure where you have individual amino acids that have been stitched together. And so you have a common backbone, but then off of this backbone, you have these things called side chains, here represented by R. And so each of these letters represents a different side chain. So for instance, valine would look like this. And if you're not used to seeing these structures, each of these kind of vertices is a carbon. So another way to represent valine would be this. just finish writing, although it's a little bit slow. But it's easier to just represent it like this. Um, so serine would look like this, and so on. So DNA you can kind of think of as a linear thing, and again, just like words, it connotes information. Protein as a functional thing, the linear ways that we can think of it as a representation, but really that linear sequence of a protein will fold up into a structure with a three-dimensional structure that's somewhat well defined that can then function. And so the molecule I'm actually showing is hemoglobin. Um, and hemoglobin is actually multiple um, proteins put together. There's a beta chain and an alpha chain, and your regular adult hemoglobin has two betas and two alphas. But basically, what's happening is this these amino acids, this string of amino acids, um, comes together into a secondary structure. So the string is called primary structure, and it comes together into a secondary structure, forming these smaller elements, alpha helices, and also these things called beta sheets. I don't think they're great examples here in hemoglobin. And then you can fold into a tertiary structure, which would be one of these, and this is kind of like just a protein on its own, but proteins interact with each other, and in the case of hemoglobin, there's actually uh, for it to function, you actually have to have all four of these things come together. This is called quaternary structure or quaternary structure. And then you can have other things that the protein's able to bind. So in the case of hemoglobin, you can actually bind this chemical molecule called heme, which is represented here, and then that fundamentally lets you carry oxygen. So hemoglobin's role is an oxygen carrier. So it's really amazing. You go from a polymer of amino acids to something like this that can now carry oxygen. This is the representation of the DNA of the beta chain of hemoglobin. So this thing right here. So now you know, if you didn't know before, all of these letters have significance. All of these letters encode for amino acids. The theme of this talk is going to be sickle cell disease, which is actually due to a mutation or a misspelling. So a change in one of the bases of DNA in the gene encoding it. So you can see there are hundreds of bases here. Um, there are many different mutations that can cause diseases like sickle cell disease, but there's one common one where this A here, this single A, has been replaced by a T. And that single change, according to that genetic code I showed you, um, changes this aspartic acid to a valine. Um, so in amino acid space, it's changing something that looks like um, this. I think I left out there's another one of these zigzags in here. Um, Changing that to something that looks like this. And that one change from something that could be charged to something that's kind of neutral changes hemoglobin so that it doesn't come together the same way it's supposed to. And that changes the shape of the red blood cell from this normal appearance, which is a biconcave disc, to this sickle-like shape. And this sickle-like shape means that blood cells that look like that can't actually get through blood vessels normally. They'll get stuck places. So instead of, you want your blood really flowing, laminar flow, getting through your blood vessels without obstruction. The second you get obstruction, you can get microclots, you can get clots. Um, and so sickle cells are tend to block flow, and as a result, cause sickle cell disease, which can be very painful. Um, can cause pain, can cause organ damage and ultimately shortens life expectancy, all from a single change in DNA. So that's where gene editing comes in. Wouldn't it be great if we could actually edit people's DNA when there's mistakes in it? And I'll be careful just to mention, we're not talking about editing germline DNA, so editing DNA that can be passed on to the next generation. We're talking about editing cells that are already um, uh, in a state where they're functioning to do something. And we're not talking about making designer babies or designer people. We are talking about correcting very specific things that happen in a set of cells in somebody. So wouldn't it be great to edit somebody's genes? So before I get into gene editing, I will stop there, see if there are any questions about what I've presented so far.
I'm not seeing any questions in chat or hands raised, so I think we'll just go ahead.
Okay, awesome. Shri, was that a question?
Thanks, Vikram. You covered the basics and the history very nicely with pictures, elaborations, and explanations. It has clarified many things to me. I just want to share a bit of context from my side. You talked about complex structures that somehow remain very stable, yet they can take on disturbances from the environment and interact with them. Like you talked about in the case of hemoglobin taking in and releasing oxygen. Temporary changes, but overall things remain stable and revert. I guess there are schemes for error detection, self correction, and preservation working in the mix. Maybe special encoding in the genetic sequences so things do not change easily, except where when needed, and do not mutate easily and in undesirable ways. So, this is not a question, but I'm sharing a bit of my perspective, which you can touch on as you go
Okay,
along.
awesome. Yeah,
Thanks.
and I think I'll be touching upon a little bit of that actually in the next couple slides. Um, so when we're talking about gene editing, we're talking about changing genes. And there are kind of two ways we can change genes. The easiest thing we can do is disrupt them. The the next thing we can do, which is harder, or took more development, is edit them. So how do you disrupt a gene? So going back to DNA, I will say that I left out the fact that DNA is actually double-stranded. And so you might have heard of the DNA double helix. There are two strands um, um, that arrange into a helix. And they're able to do that because the information is actually redundant among each strands. So a G on one strand always pairs with a C on the other strand, or an A on one strand always pairs with a T on the other strand. And actually, to Shree's point, this helps with the redundancy. So all of your DNA, you have 3 billion base pairs of DNA in 23 chromosomes, and you actually have 23 pairs of chromosomes. One comes from your mom, one comes from your dad. But for each sequence of DNA, you actually have two strands. So if there's a mutation, and this is a little bit of an aside, but if there's a mutation in one of these bases, so DNA has to get copied as you grow and as cells divide. So your DNA is getting copied all the time. And sometimes there are errors because it's not a perfect process. But let's say this C gets mutated, gets changed to a T in that process, there's still this other strand with the G here that helps tell you which one is the correct base. So there are all these repair mechanisms in the cell, and actually gene editing takes advantage of repair mechanisms to do its thing because the cell does not like to be mutated. So imagine if you could make a break in the DNA. So chromosomes are long and have millions of base pairs of DNA. but they're continuous. So let's say we cut this thing and made a break. The cell fundamentally doesn't like that. It thinks something's been damaged. And a break can lead to other things. It can lead to rearrangements in the DNA sequence. So it can it can lead to DNA sequences getting misplaced. It can lead to a lot of bad things. So you can think, if you think of the cell as a person, it really wants to stitch together these breaks really quickly. And so there's a DNA repair process called end joining that when it sees a break, it'll try to stitch the DNA back together. But it's so important, or evolutionarily, it's so important to maintain continuity of the DNA, maintain these chromosomes, that that evolution has allowed for an error-prone process in correcting these breaks. So if you make a clean break in this double helix, when the cell repairs it, some percent of the time you'll get a perfect repair, but some other fraction of the time you will get nucleotides inserted seemingly at random. You'll get nucleotides deleted seemingly at random. It turns out it's not actually fully a random process, and you can predict this, um, but you can think of it as being random. So what does this do? So remember, you're going from DNA to protein, and I said there's a code, and the code is a triplet code. So, if you remember this code, it's a triplet code. So each 3-mer of DNA encodes an amino acid. Now, imagine you deleted 3 bases in a row. That's actually a whole amino acid. This could have bad consequences, but protein structures are resilient enough that it doesn't always have an adverse effect on the protein function. It really depends on the type of change you're making. So this may or may not lead to disruption of protein function. But if instead of deleting 3 bases, you've now deleted 2, you've changed the register so that the next 3 should be ACT here, but instead the next 3 will be TCC, and the next 3 after that will be TGA. So if you look at the code, we just went from an ACT, which is a threonine, to a TCC, which is a serine. So we've changed the amino acid, that's fine. But look at the next one. We went from CCT to TGA. So CCT encodes for proline, and if you look at the code, TGA encodes stop. So in this code, there has to be a stop, otherwise you're just going to keep on going. We have roughly 20 to 30,000 genes in the 3 billion bases of DNA. And so if you didn't stop, you would just run into the next gene, and your proteins, would all be concatenameric and be really hard to deal with. So we have these stop codons. But in this example now, we've stopped, and whatever was encoded downstream of this mutation now can't be expressed. So you get a nonsense protein, or basically this protein's not going to be functional. This has been disrupted and you've knocked out its function. So all of these mutations caused by this break that you induced in the genome will lead to disruption of a DNA sequence. And I'll show you in a second how powerful disruption can be. Because you can say, why would I disrupt something? That's not actually that favorable. But sometimes, due to various feedback mechanisms in the body, disrupting something can actually cause something else to happen that you want. Or maybe you're disrupting something that was bad to begin with. So that's gene disruption. The other way you can change genes is to actually edit them, and I'll talk about this more in a few minutes. But instead of just taking a blunt scalpel and just putting things together in an error-prone way, you go in and make very reasoned changes. And could you change this T to an A? That actually turns out to be a little bit hard in this case, but can you actually edit a single nucleotide or a couple nucleotides. So that's kind of the overview of what we're trying to do with gene editing. The first gene editing experiment was described in human cells, was described 30 years ago with something called a homing endonuclease. So the next part of the talk, I don't expect you to follow completely. I'll present it, and hopefully you get the big picture part of this. But homing endonucleases are, so endonuclease, so nuclease means something that cuts nucleic acid. So this is a DNA cutter. Endo means it's cutting kind of in the inside of the sequence rather than chewing back from the ends. So this thing called a homing endonuclease is naturally found in nature. And if you Google it, it's a fascinating story of evolution. But for our purposes, it's a protein, an enzyme that can both recognize DNA and cut. So in this schematic, this enzyme, this homing endonuclease, is cutting right there and right there. So theoretically, this does everything we want to edit the genome. What it doesn't do is allow us to program it. So this thing has been evolutionarily optimized to just cut this sequence, or to cut sequences like this. If we wanted to change it to some other sequence, for instance, a sequence in hemoglobin, we wouldn't be able to do that because this is a very complicated protein that's evolved the ability to cut this specific sequence over many, 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 many years. One of the first research projects I was ever involved with was in my PhD mentor David Liu's lab to see if we could change the sequence preference of the ISKI1 homing endonuclease. And it has this long target site. And David had multiple graduate students over the first few years of his lab working on this project, other projects obviously as well. But this was a project that took a lot of effort. And the end of all that effort was changing the preference by a single base, which was really cool, but not good enough to do the gene editing that we're talking about. Because to do gene editing, you need a platform that can make changes at any gene you want, not just one sequence of the 3 billion base genome. So homing endonucleases weren't going to be the story. But what could have been the story was something called zinc finger nucleases. And in zinc finger nucleases, the recognition of the DNA is separated from the cleavage of the DNA. And so the recognition is through these zinc fingers, and I think they're called this because they kind of look like fingers, I guess. There's a zinc ion which keeps the structure together, and each zinc finger recognizes roughly 3 base pairs of DNA. And so you can array these together to recognize the DNA, and then you could put this cleavage domain to create the cut. And this is actually a staggered cut, but that's a detail that might not matter. So this was really encouraging, and there was a lot of effort to figure out how to make these. The challenge with zinc fingers is, and they're naturally occurring in the human genome. The challenge is this code isn't actually super well understood. And back in the 90s and the early 2000s, when zinc finger nucleases were in vogue, with the technology we had there, it took multiple labs, companies to try to identify this code. And today I think it's still not perfectly known. It is very well known, and we can certainly use zinc fingers and make zinc fingers that bind to DNA sequences. But there's an art to this, and there's knowledge, a lot of knowledge that's needed. So while zinc finger nucleases showed promise, and some of them went to clinical trials, they weren't perfect for this application. What seemed to be perfect was this thing called a tau effector nuclease, or TALN. This comes, the binding domains here, the binding part of this comes from a plant pathogen. And unlike zinc finger nucleases, there's actually a one-to-one correspondence. So this is a tau effector or a tail. And these two amino acid residues are actually responsible for the code. So you can see if it's H and D, it'll recognize a CG base pair. If it's N and I, it'll recognize an AT base pair. So you can imagine putting these things together. And so this was very promising. And it's a beautiful structure. Here's the DNA double helix, and these things kind of wrap around the DNA double helix. And I will mention that in general, techniques of structure determination using X-ray crystallography, which is basically expressing pure protein so pure that it essentially crystallizes, and then you use X-ray diffraction to be able to read out the molecule, the orientation of the molecules. It takes some, you need a phasing factor in there to help you solve the Fourier transforms, etc. But use kind of a diffraction-based method on a crystal to get these beautiful structures. And these kind of structures have really helped um, a lot of the gene editing research that goes on. It's an aside. But this could have really worked. But notice the timing here. This was around 2010, 2011, 2012. And Talon's got superseded by Cas9, which is the real showstopper here. So Cas9 is a DNA endonuclease, so a protein that cuts DNA. It leaves kind of a flush, a blunt cut in the DNA. And it can be programmed to cut almost any 23 base pair sequence you want with a few parameters. Or one parameter, really. The parameter is the sequence needs to end with 2 Gs in a row. And this is called the PAM, protospacer adjacent motif. This rest of the sequence is called the protospacer. This is for historic reasons. But what you need to know about Cas9 is it's a protein. So this gray thing here, these alpha helices, so these like ribbon-like things, it's a protein like zinc finger nucleases, like talons, but it's able to interact with the DNA not through the protein itself. The protein is actually a scaffold to bind to an RNA. So we usually think of RNA as information carriers, but here the RNA is what's doing the recognition of the DNA. And so you can make any RNA and the first 20 bases of it through complementarity to the DNA. So like DNA, you can have the interactions between RNAs and DNA are stereotypes. So G goes with C, A on an RNA goes with T in the DNA, just like DNA interactions. So hopefully, if that doesn't make sense, hopefully you can follow that Cas9 is a protein, and you can program it with this thing called a guide RNA. And the guide RNAs are always going to be the same. This red part here is always going to be the same. The only part that's changing is these 20 nucleotides, and that changing that 20 nucleotides is what gives you the specificity to target a specific sequence, that specific 20 bases in the genome in whatever genes there are. So this is the most important thing to understand about Cas9. It's a protein that's targeted by an RNA, and it's really, really easy to target. All you need to do is express or make an RNA that these first 20 bases are changed compared to the others. That's the only thing you need to do. Anybody can do it, and so people call this the democratization of gene editing. You can do this in research, and you can do this in therapy. This was absolutely transformational, and this was the work of Martin Yinnick and Emmanuel Charpentier and Jennifer Doudna, and Charpentier, if you remember, recently won the Nobel Prize for this work. They're the ones who first showed that Cas9 can be used as an endonuclease in this way, programmed by RNA, and that was followed up shortly by Feng Zhang from MIT, along with Jennifer Doudna's lab, showing that you could do this in human cells. So that's Cas9. And I usually talk about Cas9, but for historical reasons, we also refer to this as Crispr. And I don't want you coming away from this presentation without understanding what CRISPR means, because we, especially in the press, refer to CRISPR and Cas9 kind of interchangeably. Cas9 is a protein that's involved in a CRISPR system. So CRISPR describes evolutionary biologically a large set of proteins that are related. So here, this is kind of a representation of Cas9. Here's that RNA. So these are actually naturally occurring things, at least in some form. The form that we use therapeutically has been artificially engineered to stitch together a couple of different RNA molecules. But naturally, this is basically the same thing. So why do these exist naturally? They exist in bacteria. And actually, Cas9 that we use is called SP-Cas9 or Streptococcus pyogenes Cas9. So Streptococcus pyogenes is a bacterium, and it's actually the thing that causes strep throat. And so that's where this Cas9 was naturally found. But all sorts of bacteria have Cases and CRISPR proteins. And the reason they have this is as kind of a bacterial immune system. So bacteria can be invaded by these things called bacteriophage, which are like viruses you can think of. So they kind of inject their DNA in and they reprogram the bacteria. They cause they cause badness in the bacteria, for lack of a better term. So what the bacteria want to do is cut away this invading DNA sequence from the bacteriophage. So they want to cut it. But the first time you see this invasion of this DNA sequence from this bacteriophage, you have no natural defense against it. But if you're able to fend it off, you now have the blueprint to getting rid of it. So if you can take this DNA sequence, which is called a protospacer, and incorporate it or acquire it into your genome, then you can use this as a programmed thing in your Cas protein to cut the attacking DNA sequence and get rid of it. So to do this, bacteria have evolved this CRISPR system where you have this kind of constant structure. So this part of the molecule here, you have this constant structure repeated in the genome, and then in between those repeats are these spacer sequences, these sequences from the bacteriophage that you want to attack. And that's giving you the specificity. So in the bacterial genome, you see these clustered, regularly interspaced short palindromic repeats that correspond to the bacteriophage sequences that they're trying to get. So that's why this is called CRISPR. And then these are associated with Cas proteins, which help scaffold these RNAs so that, and then help catalyze the cleavage of the invading DNA. I'm not going to get into the whole history of CRISPR, but it involves yogurt production of all things, and it's actually a really fascinating story. Getting back to Cas9, remember Cas9 is a protein that's targeted to its sequence through this guide RNA, single guide RNA. It requires a GG-PAM, and the mechanism has been really beautifully elucidated through all sorts of structural work. What I'll say is the mechanism starts with recognizing a GG sequence, and then seeing if this RNA sequence is actually complementary to the target site in the genome that the protein is paused on. And so it's kind of nucleated near the PAM, and as it gets more and more hybridized, so as the RNA recognizes more and more of the DNA, then it basically triggers a conformational change in this protein that allows the cleavage to happen. Hopefully that makes some sense. If it doesn't, Cas9 is a protein that's targeted to a DNA sequence with something called an RNA, a guide RNA, and is very easy to do. So I've shown you how you can disrupt genes. How can you edit genes? You can edit genes by actually taking advantage of Cas9. But instead of letting Cas9 cut both strands of DNA, you only let it cut one strand of DNA. And in addition, you add another part of the protein called a deaminase to make an edit in the DNA. So remember, DNA is usually double-stranded. When Cas9 binds, those strands have to come apart because the binding is because there's an RNA that's associated, the guide RNA is associated with Cas9, and that's taking up the binding partner of one of the strands of DNA. So that leaves one strand exposed. So in the figure, what I just drew is kind of flipped for what's in this figure, but that leaves one strand exposed. So this is single-stranded DNA. And it turns out that there are enzymes called deaminases. This example is ApoBec, which is a naturally occurring protein that will edit these DNAs. These are kind of involved in RNA editing, which is an entirely different field. But you can take what originally is a cytosine, and through the enzymatic activity of this deaminase, you can turn it into a uracil. So chemically, you take the cytosine, you deaminate this, and you're left with something with a base called uracil, which is essentially functionally equivalent to T. So you can make cytosine to thymine changes, or you can make, with a different type of base editor, you can make adenine to guanine changes. And this was pioneered by my thesis advisor after I had left the lab, by Alexis Comore in his lab and Nicole Gaudelli for the two different types of base editors. To Shree's point a little bit though, the genome doesn't like to be changed, and I mentioned that with the end joining with the disruption. So you actually have to trick the genome into accepting this change. So once you change the C to a U, you'll notice that the original G is still there. So you need to trick the cell into thinking that this strand here, this U is the correct sequence and not this G. Because anytime the cell sees a mutation, it wants to correct it, because if your cell's mutated and mutated and mutated, that information content no longer provides information because it's not reliable. You know, think of a game of telephone when you were kids. If the message gets changed, and if it gets changed too many times, it's a completely different message. And this kind of deamination reaction is one that commonly happens. It is the most common DNA damage, most common DNA damage that we see. So the genome is actually trying to revert these changes so that you keep the C and don't keep the T. But our goal here is actually to change the C to a T. So in this base editing technique, you actually trick the genome into thinking that the U is correct by making a damage here. So you actually nick the genome, so you make a cut in this strand of DNA. The cell says, wait a minute, this has been cut. This whole strand is no good. We need to use this strand, and that's how you get your edit. There's some other tricks that go in there as well. This is a little bit more complicated, but I just want to say the word prime editor. David's lab, with the work of Andrew Anzalone, continued on to make a version of Cas9 that could go beyond these changes, and basically you take that guide RNA and you kind of add the change you want to the end of the guide RNA, and this acts as a template with a polymerase attached to the Cas9 to incorporate your change. It's a little bit complicated, so I'm not going to talk about it in detail. But fundamentally, what you need to know is that there are two ways to change genes. You can disrupt them using Cas9, and there are other Cas9-like enzymes that you can use, or you can edit them. So I'll pause there. I know that was a lot of molecular biology, but if there are any burning questions, I can take them now.
I don't see hands raised or questions in chat, so I think we've either lost everybody or, well, I see Siri has his hand up. Go ahead next.
It's really nice to see the levels of abstractions: molecules, proteins, DNA, genetic sequences, helical structures, etc. These make things a bit at home and comfortable for those like me coming from engineering and computer science. It abstracts the complexity of life sciences and biology and is very nicely done and gives us the hooks for understanding the CRISPR domain from purely information processing perspectives. Many things are popping in my mind, but I want to bring up one point. In self-organizing systems, there are usually ways to detect failures, damages, or errors, some form of majority voting or parity sums, etc., for deciding what is right and what is wrong. In your domain, I note that genetic sequences is encoded in two identical connected helixes. So, how do you do majority? I guess there are more built-in redundancies and other things in the mix for making things work. Again, you can just cover
Okay,
these
awesome.
things
I
as you go.
can give you a quick explanation here with the base editors. Um, there are certain types of DNA damage that are common, and that's, I think, just due to how evolution worked and how all these enzymes work. So C to T mutations are common, or sunlight will make a certain DNA lesion, and that's actually what predisposes you to mutations that cause things like melanoma. So the cell has over time evolved mechanisms to try to correct these. And that's why with the base editor, you need to trick the cell into thinking this thing is incorrect and this thing is correct. Um, but beyond that, you should think of this as an ensemble. And so you don't have just one cell, you have many, 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 many cells. And as long as, and tissues, like, think of your skin, your lungs, your hearts, you have tissues that are composed of many, many, many cells that are somewhat redundant. And so there is some protection in how replication happens. You don't want your stem cells to get mutated, which are, the cells that populate the rest of the cells in your body, because then that mutation gets propagated. But mutations that happen in cells that kind of have a very specific function and then, are going to go away and get replaced by other cells, those can be tolerated to some extent. But in an entirely different um, field than what I'm really talking about, when you do get those errors that that do confer an advantage to a cell, then you get cancer. And so that's an entirely different thing, but that's where that's evolution wasn't perfect, or wasn't perfect for what we think humans should be, have long lives, etc., without disease. And so cancer is actually really the manifestation of that error process. But you really should think of it as an ensemble. And I'll get into this a little bit in the gene editing. Okay, so I've taught you how, I've described how we can edit genes, and at least you know that you can disrupt genes or you can edit them. Um, so what is the therapy that the FDA just approved? So they approved a therapy for sickle cell to treat sickle cell disease, and the therapy is not what I've kind of set up with the beginning of my talk. It's actually different. The therapy is um, a little bit roundabout. So the therapy is not targeting hemoglobin. It's actually targeting a regulator of hemoglobin. So the genome's very complex. And so you want to turn on and off genes at different times so that you can get something as amazing as an organism. Whatever, small organisms, animals, human beings, um, there's a really complex set of events that happen, because think about it, that we all have 3 billion bases of DNA, and we all start off as a zygote, which is a union of an egg and a sperm, which is that genome, that 3 billion bases, along in a sack of proteins and some other things. But that one cell, that one fertilized zygote, leads to this complex organism. And so there are all these programs to turn on and off genes at different times. So hemoglobin is no different. And so the adult version of hemoglobin has 2 alpha chains and 2 beta chains, and the beta chain is the thing that gets that people can have inherited disorders in to cause sickle cell disease. But it turns out when you're a baby, you don't actually express that beta hemoglobin protein. You express a different one called gamma. And the reason for this is when you're inside your mother, you need to get oxygen, and you're inside, you're not breathing, you're not breathing the air. And so you need to steal oxygen from your mom. And so you need a hemoglobin called fetal hemoglobin, which has a stronger affinity for oxygen than the maternal hemoglobin, so that in the placenta, the mom's red blood cells will transfer oxygen to the fetus's red blood cells. So babies have this, or fetuses have this fetal hemoglobin in utero. For reasons I won't get into, that's not optimal once you're out in the world. So it switches to this adult hemoglobin with expression. So you can express proteins. So proteins can be turned on and off. And so you modulate how on these different hemoglobins are around birth. So if you have a mutation in this beta hemoglobin that's going to cause sickle cell disease, you're going to start producing this aberrant protein after birth for the most part. So one way to treat sickle cell disease is actually to really ramp up the production of this fetal hemoglobin, to ramp up this gamma chain. And so this program is actually mediated by a protein called BCL11A. It's a transcription factor. It's actually a zinc finger, funnily enough. But this protein, when this gets turned on, that switches the relative production of fetal hemoglobin and adult hemoglobin. So if you turn this off, you're going to have more fetal hemoglobin in your cells. And so you can actually, and this gets a little bit more complicated, you can actually target a regulatory element in this BCL11A gene that shuts down the production. And so if you disrupt this regulatory element, you're going to turn on production of fetal hemoglobin, which will treat the sickle cell disease. It's a really cool feedback loop. And if you're not familiar with biology, you might be familiar with feedback loops in engineering. Biology is full of these feedback loops. And this is kind of really cool. So there's something that, let's see if I can get this right. There's something that turns off fetal hemoglobin. So you're turning off the thing that's turning off fetal hemoglobin, so you're increasing it. A really cool technique called a Crispr screen went into identifying which sequence should actually be targeted. And I won't get into this too much, but one of the cool things about Cas9 is that because it's so easy to change the sequence that you're targeting, you can actually assess many, many different targets in an ensemble of cells. And so each of these cells, these circles, is expressing a different guide RNA targeting a subtly different sequence in that BCL11A gene. And then you can actually use instruments to identify how much fetal hemoglobin is in each one of these cells, identify the ones that are expressing the most fetal hemoglobin, and then go back and figure out what the target was of that. And so you get this this sequence here, which was the guide RNA that was the basis of this drug. So the drug that got approved for the FDA from the FDA was a combination of a guide RNA targeting this sequence plus Cas9. It's not approving all Cas9s, it's approving just for the specific indication. And if you haven't read the press, please do go and read about the benefits of this therapy. Medicine is all about weighing benefits and risks, as you probably would know from personal experience. The benefits of this approach are really life-changing. And so when the FDA recently decided to approve this, it understood that the benefits were so incredibly high. It didn't even debate the benefits. But you have to talk about risk. And for the rest of my talk, I'll talk about the risk a little bit. Um, you need to think about what could happen. Um, and so you might have seen this a few weeks ago. There was an FDA panel that said that sickle cell cure is safe enough for patients. And this has been my um, area of focus, starting with my training um, and continuing on for the past 15 years, looking at the safety of gene editing agents. Um, my son is now 9, he's in 4th grade. But when he was in kindergarten, this was actually during the pandemic, I wandered into his room one day and saw this drawing. Um, so, and he wrote, "The DNA slicer is very powerful." And he told me to, when I asked for his permission to put this up, he said something about like me referring to him as a genius or something like that. So I just said that so he can hear it later. But um, I hadn't, maybe he internalized what I talk about my science. Um, but this was pretty profound and it's been very useful for talks because the DNA slicer is very powerful. And so you have to ask the question, are you only targeting the thing that you want to target? As Sri alluded to, um, these systems, no system's perfect. My dad's in semiconductors. There are defects, to whatever parts per million, parts per billion, parts per trillion in all systems, no matter um, how well described they are and how perfect you think they are. And so in that original paper from Yannick, Charpentier, and Doudna, they actually described that you could get Cas9 to cut sequences that were mismatched at all of these red letters. And if that was the case, this wouldn't have gone anywhere. So it turns out this wasn't the case. But I'll pose a question. What happens if, in addition to targeting YFG, your favorite gene, you target something else? For instance, P53, the master of the genome. That's a risk you want to understand. And I will give you the caveat that um, the outcome we're really worried about is cancer. We're not worried about creating mutant superhumans. That's like very not plausible. It's not plausible at all. But what we are theoretically concerned about is something like cancer. Um, But I will say that cancer is a complex thing that requires more than one mutation. So I think even if you did target something like this, you wouldn't necessarily get cancer, but we need to understand it. And the reason I can confidently say that is as you age, you actually accumulate mutations in your blood cells. And in an entity that was described roughly 10 years ago, you can get what are called clonal proliferation. So a large fraction, so here's the fraction here, according to age, of your blood cells can have mutations in genes that otherwise are associated with cancers, leukemias, or myelodysplastic syndromes. But you can be relatively normal. So there are other biological consequences, which I won't get into. This is a really fascinating field. But I just want to set up that even if you have an off-target cutting of disruption of something that you think is really important, that doesn't guarantee a bad outcome. But it's something that we need to understand. Going back to this original study, I at least wasn't sure that this would be useful for anything if it had no specificity. But it turns out that very shortly after the description of Cas9, we were, as a field, able to find out that it actually was specific enough, as obviously now that we have an FDA-approved therapy, but it was specific enough to be used for this type of thing. So it's specific enough, but it's not absolutely specific. So here are some results from an experiment targeting this sequence here called the on-target, that I'm terming the on-target sequence. A little bit to Shree's question, when you do this experiment, sometimes you're not successful at actually editing a cell. You might not get the Cas9 in, so there's a delivery thing. You have to get the protein and the guide RNA expressed in cells, and that's a whole complicated thing. And once it gets to the cells, you want the cell to actually create a mutation rather than just stitch back together the regular sequence. So in this experiment, a decent amount of cells, two-thirds of the DNA from, hundreds of thousands of cells, um, um, were mutated, were disrupted in some way. But then a decent percentage of these other sequences, these off-target sequences, where these colored boxes are differences compared to your intended on-target sequence. So these are all happening at different chromosomal locations in different genes or not genes in the genome. And so you can get a set of these off-target sites. A few rules, they generally look like the on-target site. So it's kind of, you're intending to find a T here, but in a certain situation, you can tolerate an A, or a TT, you can tolerate this change, because biochemically, biophysically, while this is not a preferred state, it can still be recognized at some frequency. And that frequency is lower than the intended sequence, but it's not necessarily super low. There's a pattern to all of this, which becomes important, where the farther away you get from the PAM, the more mismatches the enzyme's willing to accept, and then the frequencies can really range. So you can get this kind of cutting, but you can ask, well, how frequent is it to get sites that look like your intended on-target site? And that goes into the information content of the genome. Genome, again, is 3 billion base pairs. You can kind of start to do the math. If you're targeting a 23 base sequence, if the genome was random, you would have 4 to the 23rd combinations, which is I don't know, I haven't done this calculation in a while. I think 10 to the 16th possible sequences, and the genome's 10 to the 9th. So generally, you shouldn't have too many things that are similar, but there's more complexity to the combinatorics, et cetera. So I'll just say phenomenologically, if you look, or empirically, more precisely, if you look at the genome, you will find sequences that have 3 or 4 or 5 or 6 mismatches. So there will be a handful of sequences that have 3 mismatches, and more, exponentially more, that have 4 or 5, 6 mismatches. And so these are theoretically off-target candidates. What I haven't said about all of this stuff is there's a lot of technology that underlies our ability to do any of this to begin with. So we can only sequence a genome because there's technology to figure out what the string of A's, G's, and T's is for DNA. We have this technology for DNA, but we actually don't have it easily for protein. And that would be really cool, but it's not there yet. But you might have heard of genome sequencing, and that's been kind of a premise underlying everything I've been saying. So you can read out the sequence of a genome, a single genome, and that's something that we can do now. And it doesn't take decades like it did with the original draft sequence. You can do it in a couple days. But to inform the ensemble of edited sites, so you see these percentages here, 66.4%. These are pretty precise. How many cells are you actually editing? So in this therapy that just got approved, the way this works is you have a person and you do all of this editing outside of the person. So this is called ex vivo therapy. You use a technique called apheresis to take some of their blood cells, which includes stem cells that you mobilized mobilized in the patient for collection. So you have a bag of hematopoietic stem cells that can become blood, and you do your gene editing, and you put this edited bag of cells back into the patient, and then that goes into their bone marrow, which is where you get the blood production. So this is called ex vivo therapy. The number of cells that go into this is, let's say 20 million cells per kilogram of body weight. The average person we learn in medical school is 70 kilograms. You're talking about 100 million to 1 billion cells. So even if you were to sequence every single letter in a genome from a patient who's been edited, who's had gene editing done, you can't actually sequence. You could do that for one cell. You could do that for some number of set of cells, but you can't do that for all 100. You can't do that for all of the tens of millions of cells that are actually edited. And that's because of cost. So there's a lot of technology that goes into this, but um, in the early days or early recent days of um, genome sequencing, the cost came down with Moore's law, which kind of made sense. Then there was some disruptive technology, but that disruptive technology has kind of played out for now, and it's roughly $600 for a single genome's worth of DNA. And this is a fascinating topic. You actually have to roughly cover each cell 30 times to reliably get a genome. But suffice to say, you can't just sequence the product. So you have to go through a two-step strategy where you nominate or screen for a set of the most likely off-target unintended sequences in the genome. And then with a confirmatory assay, go in and just at those, like, let's say 10 to 20 to 30 to 100 locations in the genome. So there's 3 billion locations in the genome, and you cut that all the way down to 100, and now you can sequence and get many, many, many copies to see if there's been a mutation. These nomination methods come with their own sensitivity floors, 1 in 1,000, 1 in 10,000. This would be like an entire another talk or two, so I won't really get into it, but just know that there's still a barrier, and we can really only say whether something has been unintendedly edited to 1 in 10,000. And the reason for that is while you get an ensemble of these insertions and deletions when you disrupt with Cas9 at the on-target site, at rare off-target sites, they tend to be small changes. And so here's a set, and this goes to reliability. This goes to here's a set of sequences. Now, if I give you the set of sequences and say, hey, I found this, what are you going to say to me? Without knowing anything, you might say, hey, well, that might just be an error. What is the fidelity of this process? Versus, is this actually an unintended gene edit? So we have a detection, not issue, but we have a detection floor in DNA sequencing, which is roughly 1 in 10,000 gene sequences. So this graph here is the editing. It's hard to see the dots, but these dots here are pretty much control dots, and this is the editing frequency. So 0.01% or 1 in 10,000. And that's basically the error rate of this process of trying to figure out what the aberrant gene editing outcomes are. And there are many ways this process can go wrong. You have to get the DNA out of the cells, so you have to extract it out of the cells. That involves chemical treatments, that involves heat. You can make mutations after the fact to the DNA. You have to amplify the DNA with these proteins called polymerases. This amplification process isn't perfect fidelity. You have to sequence the DNA, so you have to read out the sequencing, and those methods aren't perfect. In addition, you have biochemical pipelines, which are also not perfect. So sequencing, I won't get into. If you don't know about this, this is like a really cool kind of interface between engineering and technology and molecular biology. Um, um, you can sequence kind of through synthesizing nucleotides of DNA, but the kind of recent latest thing is these nanopores, where you have these membranes with a potential difference across the membrane, and you have a molecular pore that the DNA kind of gets fed through, and basically the current of that DNA is related to the sequence that goes through it. And so you can then infer what the nucleotides that went through is, and you can really sequence long stretches of DNA. It's really cool, transformative technology. The problem is the error rate for these nanopores are super high and aren't good enough for the type of gene editing outcomes I'm saying. So all of what I'm talking about is assuming all of this technology to identify gene editing outcomes is assuming that everybody has the same gene sequence. And what I really remember from the all the press around the genome sequence release in 2000 was Bill Clinton going on like late night shows and things and and talking about how we're very similar to each other. 99.9% of our genomes is the same, and I think the message was, hey, we're all really, there's a commonality to humanity. But as a gene editing outcome person, I look at that 0.1%, and that actually means we're different at 0.1% of the genome. And so of the 3 billion bases in the genome, each of us, depending on populations, so these are different populations, each of us has 4 to 5 million changes compared to the next person. That's a lot of changes. And when you're talking about gene editing, especially off targets, those changes can make the difference between getting recognized as unintended activity and not. And so one of the things that I've worked on is to take all of the information we have from sequencing genomes of people across the world to really encompass as much genetic diversity as we can, and testing to see if these sequences could be off-target candidates. And this has really been enabled by high density oligonucleotide synthesis. So oligonucleotides are short pieces of DNA. So I can call up a company or go on their website and order a pool of hundreds of thousands of DNA sequences that look like my intended on-target Cas9 sequence, and design an assay that takes into account all of the genetic variation that we know about, and assay them all at once, so that we can protect against the outcome of an individual having a rare genetic sequence that could actually cause potentially cause a bad gene editing outcome. And so this is basically like inkjet technology, which is being used with chemical synthesis of DNA. And this has really been transformational for my work. The other thing that we can do to assess the contribution of just natural genetic variation in the population. So again, certain genes people will have that have different spellings from everybody else, is using computational methods. And these really came to the fore in the last few weeks, as the FDA was deliberating, because a computational method that's published has identified a sequence that's a genetic variant in somewhere between 1 to 10% of African genomes. So if you sequence people from Africa, different populations, in roughly 1 to 10% of some of those populations, you will find a difference in the DNA, which makes a difference between an off-target site that can get cut by this Cas9 that the FDA was considering, versus not cut. So this is a bona fide off-target in individual people's cells, and a computational method to study off-targets was able to identify it. I will say that the method I mentioned using those DNA chips that I'm developing has also identified the site in unpublished work. But this was a lot of the focus. That study that I just mentioned, this computational study, was a lot of the focus of the discussion of the risk of gene editing for this particular target site. For every different target site, you have to assess the risk separately, because even though it's the same platform of Cas9, it's a different sequence you're targeting biochemically that leads to a different set of a different risk set. But it was a really fascinating conversation, and it'll be hard to get this link, but I urge you to Google this. This was the name of the drug, which I'm probably not supposed to say based off of what my institution wants me to do in these talks. I have no relation to this particular drug, though. But Google this. The video is on YouTube. It's it's a day-long meeting, so it's a lot of stuff, but fascinating patient testimonials, expert testimony, and hopefully after listening to this presentation, you have some framework to understand this deliberation, which was really cool to see. So what does this computational method do? I have some experience with these types of computational methods, and I'll say with zinc finger nucleases, we basically profiled many different potential sequences that could get cut, and we applied a naive Bayes classifier to take all those sequences and come up with a classification score of which ones could actually be cut, so a probability score and we're actually able to predict off targets that way. The thing that underlined the computational method that I just mentioned is called the CFD score, cutting frequency determination, um, um, uh, created by John Dench and others. And that used kind of a similarly large data set to distill what could be predicted as off-target recognition and cleavage. And as Cas9 and gene editing get used for more and more and more and more things, this will be very important to be able to take that 3 billion sites in the genome and cut it down to a reasonable number of things, taking into account genetic variation. And so the method I mentioned with the oligonucleotide chips is one part of this. Computational methods are another part. But I will mention about the computational methods that there's still a lot of room for improvement. They are not super sensitive. So they're really, really good, but they're not perfect for this application. And this is where big data, where machine learning, perhaps AI, could make a lot of inroads. So I'll come back to this. I will say in work that I won't, since we're kind of getting late on time, you could say, well, could you make Cas9 more specific so it doesn't actually recognize things it's not supposed to? And so there's been a lot of work that's gone into this. And it turns out that you can make actually changes to Cas9 itself to make it more specific. You can take insights from structural studies to help make it more specific. So you can do all of that stuff, but the FDA approved therapy is actually just a regular Cas9 that comes basically straight out of Strep pyogenes. That's good enough and specific enough for this particular sickle cell application. Other work that's kind of ongoing that's really cool, which also involves machine learning, is to try to identify other Cas9-like things that could be useful. So there are some limitations to Cas9, and there's such a large diversity of these CRISPR-Cas proteins in nature. There have been many efforts, many computational efforts to try to identify them. I will just highlight one that just literally got published in the last couple weeks out of Feng Zhang's lab. I'm not going to claim to understand any of this stuff, but basically this was a large scale effort to study genome sequences of bacteria to identify things that could be CRISPR and and guide RNAs and put them together and identify new Cas9-like proteins. And there's a lot of, there's a lot of potential with this type of genome mining approach to create new revolutionary things. So I apologize that I went through the last part quickly, but the summary of the last part of the talk is basically that to get FDA approval of a drug, you need to make sure that your very powerful DNA slicer is not so powerful that you're exposing people to risk. It's hard to just directly read out the unintended side effects, so to speak, the off-target editing by Cas9 and other gene editors. So you need kind of a phased approach, and you also need to take population diversity into account. So that's kind of my overview of Cas9. I described a therapy from the FDA that involves what's called ex vivo therapy. So this was taking the cells out of a patient and putting them back in. There's another thing you can do to edit people's genomes where you actually deliver Cas9 to their cells directly inside of them. And this would be in vivo gene therapy. The liver is a great target. This is not my expertise, but there's a lot of technology that needs to go into targeting things like lipid nanoparticles, carrying purified Cas9 proteins, or carrying the DNA or RNA sequences that express Cas9 to the liver to correct things, metabolic disorders like phenylketonuria, things of that nature that primarily are disorders of the liver. And so that's another challenge that there's evolving technology, but is very much on the frontier of opening up the possibilities of what we can do. So with that, apologize for talking for 80 minutes or whatever, but hopefully this was informative. And if you have specific questions, I'm happy to take them now, but also, here's my email address. And if you want to talk more and you have some crazy idea and you want to just talk through things, feel free to reach out to me, and I'd be happy to engage with anybody and, talk more about, this kind of exciting technology.
Well, thank you very much for a detailed and fascinating discussion. I knew a little bit about this, but definitely picked up a lot of new and interesting points. Obviously, there's a close correlation, as Siri had already identified, with the kind of things we think about.
Jim, can you hear me? I just want to check if anyone else wants to ask a question or a comment. So give them a few minutes. Just want to give others a chance. Okay. I just wanted to share some more comments from my side and possibly also ask a question. Vikram, this is quite fantastic, especially for people coming from and living in the world of information processing, automation, and autonomous robots. It gave me a good high-level understanding of how things are working in the CRISPR Cas9 gene editing processes. I view them or see them as autonomous nanorobots in action, released in a living host or in a lab-hosted environment. And they are programmed in a variety of ways, no centralized control, but working like an ensemble in a distributed, coordinated action. And he also talked about various schemes for effective feedback and control for tackling scale, complexities, and uncertainties. I see lots of applications of these ideas in other domains, especially the domains where computer scientists and engineers have to really support or innovate and make things possible. I'll touch on one of the critical challenges we face today, which is the consequences of fakeness. If we cannot solve this problem, soon all our civilizational order will come crumbling down, as we will not be able to trust anything. I think there should be productive ways from biological domains, especially gene editing, to stamp digital artifacts with authenticity for detection of fakes and also for some level of robustness. Hope you can address some of these things before we wrap up. But I just want to say in closing, hope you can come back again to continue what you have opened up today. There is so much need for cross-domain education and synergies. This is not just for IEEE New Hampshire, but for all of IEEE. Engineers and computer science, the core of IEEE, can significantly benefit from the approaches to tackling complexities, scale, which underpins your domain, and synergize them for challenges in other domains. So I thoroughly enjoyed your talk and presentation, and thank you so much, and look forward to seeing you again. Thanks. Bye-bye.
I want to thank everybody for joining us. Put a note up, which I will, in the chat, but we have a certificate of appreciation and a thank you gift that Siri and I will make sure get passed along to Dr. Pat. And then we'll make sure that this also gets put up in an appropriate way on the IEEE New Hampshire YouTube site once we've had a chance to do an editorial pass on. Thank you all for joining us, and thank you so much for a really informative discussion.
Awesome. Thank you for having me.